Hi, my name is Karen Morris, and I'd like to tell you a little bit about my research that I did as part of a collaboration between Hendrix and OSU over the development of a drug called HOSU3, which is a novel dihydroorotate dehydrogenase inhibitor specifically for acute myeloid leukemia. So a little bit of background on what leukemia is. Leukemia is a cancer of the blood. So your normal blood cells are made in your bone, specifically your bone marrow, in which stem cells mature into your mature blood cells and eventually go into your bloodstream. But when you have leukemia, these stem cells don't fully mature into the blood cells. Instead, they get stuck at this state of immaturity, and they pick up a mutation for rapid division, filling up the bone marrow. As they fill up the bone marrow, eventually they start to leak into the bloodstream, causing leukemia. So the World Health Organization states that acute myeloid leukemia occurs when about 20% or more of these immature blood cells are in the bloodstream. So the main reason we want to study acute myeloid leukemia is because the number of cases has increased significantly during the years 1998 to 2018. So acute myeloid leukemia has increased by around 10,000 cases. CLL, or chronic lymphocytic leukemia, has also increased around the same amount, but the difference can be seen in the number of deaths during the same time period. So acute myeloid leukemia has increased by around 4,000 deaths, while all the other subtypes seem to be either leveling off or decreasing in the number of deaths. So the likely reason for the increased number of deaths due to acute myeloid leukemia is because there's a lot of mutations that can lead to acute myeloid leukemia, and these mutations can often lead to relapse in patients or resistant to the current treatments that we have. So the current way that we treat leukemia is very different than how we treat solid tumors. So the main line of defense for solid tumors is to surgically remove the tumor and then treat those remaining cells with radiation, but this doesn't really work for leukemia. So our main line of defense for leukemia is to use chemotherapies. We can also use radiation and bone marrow transplants, but those are a little bit more difficult. So recent drug developments with chemotherapies have found inhibitors that have caused these stem cells to mature into the cells that they're supposed to be from this immature state. So that's really exciting news, meaning that these immature cells are going to the mature cells they're supposed to be. But the main thing with most of these new inhibitors is that they're only focusing on specific mutations. So our goal was to develop a drug that targets all or most of the mutations that lead to acute myeloid leukemia and making these immature cells mature into the type of cells they're supposed to be. And a way that we want to do this is through targeting an enzyme called dihydroorotate dehydrogenase. So this enzyme is part of the pathway that makes nucleotides. So a little bit overview of how we developed our drug and got it kind of ready for clinical trial was that we looked into its effects on cellular cultures. We tested it for signs of differentiation, which differentiation is basically just the maturation of those immature cells into the cells that they're supposed to be, and we investigated its effects on a living system. So specifically, we used mice. So our first step was to look on how it affected cellular cultures, in which we used an MTS assay. So basically what we did here is we want to see if there is any concentration and at what concentration our drug caused 50% of our cells to die. So what we did find is that HOSU3 did kill at least 50% of our cells in the cell culture, and it needed at least less or a similar amount to an already known dihydroorotate dehydrogenase inhibitor called Brequinar. So this is exciting news, meaning that we can move on to our next set of experiments looking into differentiation, or seeing if they're maturing into the cells they're supposed to be. And the way that we did this was through an imaging assay, looking for the morphological changes. So up here is our negative control, so what we expect to see is immature cells, and that's what we do see. So we can see a large amount of nucleus and a very little amount of cellular body in most of these cells, which is showing an immature myeloid cell. But when we treat with HOSU3 and Brequinar, but specifically I want to focus on HOSU3, we see the breaking up of this nucleus, which is showing a neutrophil. So a neutrophil is a type of mature myeloid cell. So that's exciting that it does seem to be showing signs of differentiation. So our last step, now that we have those two, is to test and see how it works in a living system, in which we used mice, and we had given our mice leukemia, and then after 3 days of developing their leukemia, we treated with our drug, HOSU3. And what we had found in comparison to our vehicle or our control mice was that mice that were given HOSU3 did live a significantly longer time period. So this is exciting, meaning that it does seem to be working in living systems. So our conclusions is that our drug HOSU3 causes differentiation and cell death in cell cultures, and it has a positive outcome for both cell cultures and living systems, meaning that it's maybe a viable option. So our next goals is to work on lead optimization studies, and this basically just means working on the best chemical composition of our drug to lead to the best efficacy or the best living outcomes. And then our next thing that we want to investigate is possible combinations with our drugs through 2 different methods. So we can do either the pharmacological approach or a genetic approach. So I would like to thank the James Comprehensive Cancer Center, who helped me throughout this project, Dr. John Bird and his lab, and Dr. Aaron Hertlings, who group I was part of, and Dr. Ola Elgamala, who is my mentor in working on this project with me. And then also at Hendrix College, I'd like to thank Dr. Goodwin, who is the connection between OSU and Hendrix, and Dr. McDonald, who is my mentor. Thank you.